car, Roger.
So, all right, next it's my distinct pleasure to introduce Dr. Conwit. She's a neurologist and program director in the Office of Clinical Research with extensive experience in clinical trials, neuromuscular disease, and clinical neurophysiology. She's also an NINDS project scientist for Neuronex. Prior to working at NIH, she was a neurology department faculty member at Johns Hopkins, subspecializing in microbiography and neuromuscular disease, with clinical trials experience in ALS and diabetic neuropathy. Her prior experience also includes running an ALS clinic at the University of Pittsburgh, where she was the principal investigator for ALS clinical trials. Dr. Conwit earned a bachelor's degree from Colgate University, where she was a Pi Beta Kappa graduate, magna cum laude, attended medical. Robin is also one of our course directors, and it is just my distinct pleasure to have her talk to us further on the data and safety monitoring course and read more about her activities.
Thank you. So today we're going to talk about boards or committees that objectively evaluate clinical trials to make sure that they're conducted in an ethical and valid manner. In 1967, the External Advisory Board first recommended that such committees be created in order to monitor safety, efficacy, and the conduct of clinical trials. I have nothing to disclose. I'm a thorough government employee. And back to the DSMBs. The degree of oversight is mandated for sufficiently complex trials. That is when the stakes are high enough. How many of you have experience with data safety monitoring boards or safety monitoring committees? Okay, great. So those of you know somewhat of how it proceeds. I'm going to describe primarily the way NINDS does their data safety monitoring, but I know there are variations in the industry and other institutes, so feel free to comment at the end about your experience. So the data safety monitoring board essentially is an NIH-sponsored mechanism, and the roles of the DSMB or safety officer should be to ensure fidelity to trial protocols and prevent a knee-jerk reaction to noise in the data. So there's ongoing review of the data by an independent individual, either a safety officer in a smaller study or a committee, the DSMB. This ensures the trial can continue without jeopardizing patient safety. It ensures the trial is conducted according to the highest scientific and ethical standards. Hopefully ensures high quality, validity, and scientific integrity, and assists in determining if any of the treatment procedures should be altered or stopped. There's always some risk involved. Hence the need for minimizing avoidable risk as Roger described. One risk is that new information becomes available in the course of a trial, and the DSMB can advise on whether to proceed, how to proceed, and how to inform participants in an ongoing trial. This is something we've encountered before. The responsibility is that DSMB members report only to NIH, not to anyone else, and not directly to the clinical investigators or anyone else. NINDS is responsible for oversight of data and safety monitoring to ensure safety and quality, timely and effective monitoring, that those have adequate expertise, and that the Institute is informed of recommendations that emanate from monitoring activities. Who is on DSMB? Well, maybe some of you are interested in participating in DSMB if you haven't before. If so, give me your names and I will try to help you. Often try to encourage young junior people to participate and get their feet wet, so to speak. So the key person is the chair of the DSMB. This person should be an opinion leader and also able to balance opinions of the other members. There must be a statistician as well as a chair who understand advanced statistical methods. Also relevant clinicians, methodologists, and sometimes an ethicist or increasingly a patient advocate are part of DSMB. Members must be completely independent of the investigators and have no financial, scientific, or other conflicts of interest, and they have to attest to this in writing each time. The safety officer. So at NINDS, the good is we have a committee that informs you what level of monitoring your studies need. It's a very small study with minimal risk. PI, the IRB, and NINDS safety officer are sufficient to monitor the study. But for a larger study, or when there's more than minimal risk, we involve, first of all, a safety officer. And in small single-site studies, again, safety is often performed by a statistician in conjunction with a safety officer. This person is appointed by the granting institution, and we use adverse events and serious adverse events on an ongoing basis to determine if the action is needed. For NINDS-sponsored study, which actually entail more risk, an independent medical reviewer is often appointed by the, usually the data coordinating center, to review adverse events in a real-time manner. Sometimes the clinical coordinating center or the PI chooses this person. This person also should be independent and not even be employed by the same institution. If there's anyone who's involved in the study. So as you can see here, NINDS is the center of the universe. And the DSMB is appointed by NINDS and advisory NINDS. The NINDS DSMB liaison is a person within my group in the Office of Clinical Research who assists with production of information, dissemination. And that person facilitates all communications, so the PI is not to speak directly to the DSMB. And the funded clinical trial, of course, is NINDS funded in this model. So what are the DSMB activities? Well, they include performance data, looking at recruitment rates, dropout rates, retention rates, safety reports, treatment monitoring, nerve analyses, as well as stock numbers. The DSMB members are also responsible for protecting confidentiality. I have to stress how important this is. Confidentiality in these cases is paramount importance. So keep that in mind. If you ever agree to participate, there should be no leaks. So the DSMB members also have to decide whether to continue or conclude a trial, monitor recruitment rates associated with ensured goals that will be met. And also, there's a question, is it feasible? Sometimes a trial goes on so many years, it's no longer feasible. Or maybe clinical practice has changed, it's no longer relevant. So these are all questions that the DSMB can evaluate. And the DSMB can request to see all blinded and unblinded data. They can request reports at any time. And they can and have advised the Institute to start, stop, or modify a trial protocol. So I was just going to bring up one point about sometimes there's a low event rate. On a past grant, we had a low event rate, and there had to be a sample size re-estimation. And so that meant more participants were exposed for a longer period of time. This is quite common now in stroke studies because stroke treatment is getting better and better, so that often sample size re-estimation is part of the analysis that the DSMB does. So who is part and how does this work? So one difference between the open and closed session is who participates, and the other one is, what is discussed. So in the closed sessions, they talk about patient accrual, compliance with protocol, as well as problems encountered. In the closed session, unblinded safety and efficacy data is looked at. That's the big difference. The course is very confidential. And then the session will discuss whether to start, stop, or modify trial protocol. So in terms of the general overview, this does change from time to time. There's often first a closed executive session with the DSMB members and the NINDS and DSMB liaison only. And then there's an open session that includes the coordinating centers, principal investigators, institutional staff, the DSMB members, the liaison, and the NINDS program directors, which is myself. Then there will be a closed session where the DSMB members and study statistician is added, and the DSMB liaison. Finally, an open session where everyone comes back together to hear the determination. well as establishing monitoring guidelines. And thereafter, usually there are biannual meetings, twice a year, either a conference call or preferably one face-to-face. They review complete safety data, and if appropriate and interim analysis is conducted, but usually these are pre-specified, and only if there's a reason to do another one is there need. So, and that leads into these emergency meetings that are sometimes called by the DSMB or NINDS at any time, especially if patient safety issues arise. So, Dr. Lewis is going to discuss 3 case studies, and I will stay here and add my 2 cents. No one does it quite as well as Roger in describing these cases, and I'm eternally grateful for his help.
Well, wait till you see where the stories come from. I do have good stories, but these are also good stories. Yeah, my financial social change. So I have the enviable position of standing next to Dr. Conwit and describing cases that she described as the first offer for publication. And I don't actually remember how we ended up with this arrangement. So I do use this for everything. Never often good week placebo. Okay. So the first example, and there is a theme to this, but I'm going to tell the stories and come back to the theme at the end, was the experience with North American symptomatic carotid endarterectomy trial. And how many people here know about this trial? Okay. So this was a trial in patients who had recently had a recent adverse cerebrovascular event and had ipsilateral carotid stenosis that was incomplete. And we divided them into 2 groups, 30 to 69% stenosis, 70 to 99. And they looked at those 2 strata based on the degree of the carotid stenosis separately. And they were trying to figure out if medical therapy versus endarterectomy was better for preventing subsequent ipsilateral strokes. And here's a report from New England Journal in 1991. So they report the results of 659 patients in the latter stratum, which is the higher degree of stenosis, who had a hemispheric or retinal TIA or non-disabling stroke within 120 days before the procedure. And what they found was that the cumulative risk of an ipsilateral stroke at 2 years was 26% in medically treated patients and 9% in the surgically treated patients, which yielded a highly statistically significant reduction in risk. And they reported that so that this information was immediately available, depending on the period of time, but immediately available to inform therapeutic decisions in patients who fell into this category. So how did that happen? So the trial was originally powered to detect a 50%, so a smaller reduction in the risk of stroke in each of the 2 groups. And there was supposed to be 600 patients with 5 years of follow-up. The stopping rule, the early stopping rule for the trial was that there was a statistically significant difference that was highly significant in double O, and that p-value persisted for 6 months. Not a rule I think we would use in modern times, although occasionally I see rules like that. And it also included a futility rule, so if the trial was trending towards futility, they weren't able to show a difference, they could also stop early. And they stopped early within that strata with about 1.5 years of follow-up instead of the planned 5 years of follow-up, so saving 3.5 years of getting this result out. And that, as I mentioned, the clinical alert was quickly circulated to physicians. So this is an example of early stopping for larger than expected benefit and allowing you to get the information out to clinicians. Here's a different example, and just to show that I'm somewhat fearless, we have the first author of the audience. Okay. So this was a study that determined if long-term topiramate therapy is safe and slows disease progression in patients with ALS, double-blind placebo-controlled multi-center randomized clinical trial. Participants with ALS, about 300, were randomized 2 to 1 to receive topiramate. So what does that 2 to 1 tell you? People were really hopeful. They wanted to maximize the potential benefit that there. or placebo for 12 months. The primary outcome was the rate of change in upper extremity motor function. And what happened? What happened was they found that patients treated with topiramate showed a faster decrease in arm strength during 12 months that was statistically significant at p.012. So this is not only a statistically significant result, but it's in the direction of harm. And the trial, well, whether it was stopped early depends on how you look at it. So the plan was to take the patients who were randomized to the active arm and include an open label continuation phase for those patients. And this design really captures the optimism of the trialists and the time they were designing before reality intervened, in believing that this was going to be good and we were going to give people who participated in the trial a chance to have continued benefit from the therapy. Is that fair? Okay. And by the way, this is distressingly common, that people are really optimistic at the beginning, because if you weren't optimistic, how could you sacrifice years of your life to get one of these things run? And you know, the Jeff Savers of the world were willing to fail over and over and over again, not that time. And it's amazing how frequently that optimism later, when you look back at it, was completely well-intentioned and based on good thinking, but how often it turns out that the reality is quite a bit different. So at the end of the randomized component, before they did this open label extension, the DSMB recommended immediate termination of the open label phase based on, if you read the publication, it says based on an excess number of cases of thromboembolism, 12 cases, 6% versus 1 case. Remember, there was twice as many people on the active arm as the control arm, which trended towards a statistically significant increased risk of thromboembolism in the treated patients. I originally had on the slide, and I've just left it there so you can see that a faster decline in strength didn't drive early termination of that open label phase, because that's what I concluded reading the publication. It was later pointed out to me, and I think this is the thing that they pointed out, which is that actually this was taken into account. I just couldn't tell from the publication. And then later it was communicated to me in the elevator this morning that the DSMB apparently was put together quite late in this trial. And they weren't put together prospectively. And one of the things that's critically important we now realize is that the DSMB is put in place with the charter, a clear understanding of what their decision parameters are, what they're allowed to make recommendations on, the data on which they're going to base those recommendations early on, so they have plenty of time as the information starts to come in to think about it. It also illustrates the fact that the reporting of DSMB activities has really changed. There's lots of trials and literature that are stopped at sample sizes other than the originally planned sample size, and you cannot tell what role the DSMB had in that early termination. And luckily, the reporting of clinical trials has gotten much better. It's much easier to understand what drove those terminations. Another example. So this was a trial in thousands of patients who recently had TIA or retinal ischemia, and it was a comparison of aspirin to clozapine, an alternative antiplatelet agent, to prevent recurrent events. The 3-year event rate from non-fatal stroke or death from any cause was 17% for clozapine, 19% for aspirin. Now, for most of us, 17 and 19 seems close, which they rewrote as a 12% risk reduction, because that sounds a lot better. And then what I really like, and the statisticians I think will appreciate that, which gave us a 95% confidence interval from -2 to 26%, which seems to cross the null, but magically they got a statistically significant p-value of 0.048. Okay? Obviously there's a reason I'm showing this example. The number of statistical examples that go like this: marginally statistically significant results, focus on a subgroup, launch a new trial, and it turned out you were fooled. That is a very frequent series of events. So what happened? So they concluded here that the clozapine was somewhat more effective, I like the somewhat, there's an edge word, in preventing strokes in the population. So someone went back and did a subgroup analysis, and they looked at the non-white patients in that original study and found that the apparent benefit appeared to exist in that subpopulation. So this is an unplanned, as far as I can tell, is an unplanned subgroup analysis. It's a little hard to tell from some patients. Okay. Correct me if I get it wrong. Okay. So then someone puts all the effort to do a new independent randomized trial in African American patients, comparing. And not only do they find that there is no evidence of benefit of clozapine over aspirin, the actual point estimate is in the direction of harm with a hazard ratio above 1, suggesting increased risk of recurrent events with the active treatment. This essentially shows that marginally significant event, initial result, may or may not have represented a real treatment effect. The subgroup analysis was almost certainly misleading, and the conclusion was that the new agent was better. But the DSMB thought process is interesting. So the DSMB was doing a futility analysis where they were asking the question, should the trial be continued to the point where, should the trial be continued because there's a realistic chance of demonstrating the superiority of clozapine versus aspirin in this population? And after 80% of the planned number of events have been approved, they calculated that there was a less than a 1% chance that clozapine would be superior to aspirin, and a 50% chance that aspirin would actually be better. So at that point, the thought process is, we are not going to prove what we set out to prove. If we continue, we might be able to show aspirin is better. The question is that important? Well, diclozapine had a much higher side effect profile, some of which were quite serious. So proving that aspirin is better than something that is dangerous, okay, isn't really that much of an advance in science. So the DSMB recommended termination of the follow-up based on the cost, and since proving the superiority of aspirin was not deemed relevant. The directions of the conclusions you could draw are fundamentally asymmetric, because aspirin had a well-known safety profile and was as effective. So what are the general conclusions here? One is, and I'm really quoting a clinical pharmacologist at my institution who's been saying this for years, which is, you never know as much at the beginning of a clinical trial as you think you do. The problem with this statement is as you start, if you believe it and you think, yeah, maybe I don't know as much, you're still wrong. Okay? It is amazing the things that you think you understand that are going to pan out differently. So the DSMB is put in place so that as reality starts to accrue through the data that are gathered, you're in a position to react to that in a way that preserves the ethical and scientific balance of the trial. For the DSMB to do this, they are always balancing safety and efficacy. So the DSMB should always have access to both data. It should always be looking at unblinded data. And one of the things that's in Robin's slides talks about how the DSMB can always ask for unmasked data. My personal opinion, and an opinion that's in the new recommendations for DSMB conduct by the Clinical Trials Transformative Initiative, the CTMC Aug people, is that DSMB should always just look at unmasked data. It is a waste of time to ever blind the DSM. Okay. The DSMB also should monitor futility because it's never ethical to continue to expose patients to risk if there's no meaningful chance of moving science forward. They should ensure fidelity to and the appropriateness of the original trial design. Fidelity to, so that at the end of the trial we can draw conclusions with appropriate statistical rigor. Appropriateness of, so that if something new happens, a new treatment becomes available, okay, penicillin for syphilis, they ensure that the original trial design continues to be ethically appropriate and should continue. And if the DSMB does this job well, they minimize the risk to human subjects, and they help us assure that resources, whether they're industry resources, federal resources, foundation resources, are well spent on the clinical trials. So I'll stop there and I'd be happy to entertain any questions. Okay. Okay, so the burden of adverse event reporting to DSMBs is a very fluid area. It depends on the risk profile of the study. So there was a period of time early on where the thought was that the DSMB should see all adverse events, usually some sort of, for just simple adverse events, some sort of counting by category, measure category, or some other categorization system, and they would sort of look at them all. And then serious adverse events would be reviewed in a hopefully a more timely fashion and as individual events. There has been some accumulated experience over the years that suggests that just looking at adverse events, especially in trials of patients who are substantially ill or injured, is actually not very effective as a way of improving safety. So I was chaired a DSMB for a large sepsis study, and I mean, every patient had many, many bad things happen to them. And what all we cared about were the serious adverse events, the so-called USARs, that were also unexpected. So if all the usual things happen to patients with severe sepsis, we didn't want to pay attention to that. So the charter should lay out what events are reported, how they're tabulated, how frequently they are looked at.
I just wanted to add that the medical safety monitor looks, like the independent medical reviewer, looks in real time at these. And if he's alerted to something, he will contact you. So there is a double.
Right, and in an industry setting, there's also an equivalent medical monitor who looks at everything. Those people often they have more than one trial they're looking at. They're sort of a single paper or the electronic equivalent. Whether or not they see patterns, it's a tough job.
I would say NINDS
Studies are monitoring at the same time, so it's a little easier to see that people who do it, perhaps it's harder to bigger trials. But there still is somebody looking over the data at the time.
I would also say about that, I can see that the medical monitor is here to pick up if a single event is so distinctive and insightful, it should raise attention, and they don't have to recognize patterns, whereas the DSMB recognizes the pattern.
Hopefully. I think that's exactly right, Rob.
So a question about that last example of the aspirin study. So you didn't say for sure, but you implied that this discussion of whether to go for the two-tailed or one-tailed futility was something that they decided when they confronted that situation. Really, that should have been defined by the stopping rules before they started. If they had created a stopping rule at the beginning that was two-tailed, and then got themselves in that situation and said, no, that was dumb, we should have done one-tailed. What do you do with the tension between the DSMB having the flexibility to do whatever's right at whatever time they want and following their own pre-specified guidelines? Because it seems like there's always a chance for bias to get in, and that's why I'm surprised about what you said about the masking.
Okay, so what time is this supposed to end? That's not because I don't want to answer the question. I just want to make sure.
Yes, 10 minutes.
Okay, so as always, Rob, you varied several different things in that question, so we try to handle it. So the first, the criteria stopping for success and futility are never symmetric. I cannot picture a situation, maybe you can come up with a counterexample, where you want to prove something new is good with the same assurance you want to show that something standard is good. So they're fundamentally asymmetric. So all of the clinical trials literature that talks about two-tailed testing is stupid. Okay? And when you talk to the FDA now, they don't talk about two-tailed 0.05. They talk about one-tailed 0.025 because they don't need the sponsor to prove their drug kills people. That's just not a useful thing. So the stopping rules are always asymmetric. The stopping rules that around which you have to have the most pre-definition and rigor are the early stopping rules for success, because that's what allows you to define the type 1 error risk for the trial. That's why we have O'Brien funding, stopping rules, or alpha spending function, or all of these mechanisms to make sure we understand from a frequentist point of view the type 1 error risk under the null hypothesis. That's all for positives. When you have an early stopping rule for futility, by definition, if you act that rule, you're not going to claim success. So that activity cannot increase the false positive risk for the trial. It cannot increase the type 1 error risk. So there can be inherently more flexibility around it. That said, I think that a DSMB should always think ahead of time about what kinds of patterns in data and what kinds of analyses would support early stopping for futility, because we have an ethical and societal and fiscal responsibility to not throw good money after bad on patients at risk, but there's no meaningful chance of benefit to them as individuals or to society. For me, the most useful metric for understanding futility is the probability at the time you're looking at data partway through a trial that at the end of the trial, that trial will be positive. So that this is a predicted probability. Sometimes you can call it conditional power, but the way conditional power is calculated in the literature is often not correct. But the predicted probability. So if I'm on a DSMB and halfway through the trial, I know there's a 10% chance that trial will be positive at the end. That may or may not justify stopping early for futility. If I'm working in an area like pain control where it's just a pain reliever, that may be a very small conditional probability and I may stop for futility because it's bizarre that it's not working. If I'm working in a disease-modifying area for Alzheimer's and I still have a 10% chance of success, that's better than anything I've ever seen, which continues. The context is really important. But to me, that's the metric that's useful. But a key idea is that the DSMB can be more flexible in futility criteria because the variability in what they do doesn't affect the type 1 errors. The only exception to that is that if a pre-specified futility rule was included in the calculation of the type 1 errors, then ignoring futility can't be, so not doing it, but ignoring futility can increase the type 1 error risk. So the FDA now commonly asks, if your design includes a futility rule, to tell us what the type 1 error risk would be if there were no futility rule followed every trial to the end. So there's a fine point there. Did I miss any part of your question?
I think we should let other people go.
Okay, Chris.
So, be kind of following up on this, one of the challenges with this, because as you mentioned, it's complex in terms of what DSMB do. And DSMBs are like study sections. They're different personalities. They all take on a different, paradigm. I wonder if you could comment a little on one of the challenges that we've run into a number of times is you may not agree with the recommendation that DSMB makes, but if the sponsor accepts it, you kind of have to live with it. But when you go to publish the manuscript, often you have to defend the decision the DSMB made that you may or may not agree with. So the reviewer may raise criticisms that you absolutely agree with, but you have to defend to get the paper published. I wonder if you could comment on that.
Okay, it's a great point. So the DSMBs always, don't have decision-making capability. They make recommendations. And they make recommendations to the sponsor, so the problem is very clear that the sponsor for NINDS studies is NINDS. That's why the DSMB reports to NINDS. The DSMB is in the position of seeing unblinded data, and in many cases, once the sponsor sees unblinded data so that they can understand the basis for the DSMB's recommendation, they have already essentially ended the trial from a regulatory point of view. That's in a for-profit or FDA-regulated setting. In NINDS, it's a little different. I think you could ask to see unblinded data, understand the recommendation.
I know it's usually at the end point, so there's a good correlation there. But it's hard to go back to try to leave some people still blind so that not everyone is blind.
Yeah, so it's a really interesting problem. So the way you mitigate this risk is through selection of the members and especially the chair for the DSMB. And the thing that scares me the most is when people who select DSMB members are motivated by things that probably aren't well correlated with selecting good DSMB members, like the PI for an academic study picks his or her friends. Like in industry, the sponsor chooses people they see as key opinion leaders in the therapeutic area because they think their drug or device is going to be successful and they really want that person to be on the list of people who thought it was good. The thing that makes a good DSMB chair or member is someone who's willing to spend a lot of time understanding the data, who understands statistical, regulatory, and clinical issues and can integrate them. And the point that Robin made, which is just so important, is the chair needs to be someone who can hear the different points of view and integrate them and doesn't just run the discussion. I guess it's true of all committees, that if you have a committee whose atmosphere encourages the expression of minority viewpoints and understanding of minority viewpoints, it will work well, and you get the wisdom of the group. And if you have a committee that's run like a dictatorship, it may not get the right answer. But I think the sponsor or the principal investigator is at the mercy of the wisdom of the DSMB, and so choosing people who understand this kind of decision making is really important. And you want the DSMB to have a very high probability of making a decision that you can actually easily defend in publication because it's clearly the right decision. I've yet to see a sponsor not go along with the recommendation of the DSMB, reflecting the fact that what can they do? They don't know the basis for it, so they just kind of have to go with it. Other questions, comments? Okay, great. Thank you very much.